Hello everybody, welcome to the week 8 video for the Kombucha lab. In lab this week, you're going to be doing what's called the TTC assay. The TTC assay measures how much acid was produced in your kombucha jars. And so there's two types of response variables that we have been measuring in the kombucha. Response variable number 1 is yeast concentration, which is colony forming units per milliliter. If the change that you made to the jar is beneficial, then your yeast colony forming units per milliliter should be higher. If the change that you made to the jar has somehow adversely affected the microbes, then your yeast colony forming units per milliliter should be lower. Right? Hopefully this makes sense to you. But the second thing that we're going to measure, which we've been saving up to do, and we're going to do this week, is the amount of acid produced. So the bacteria that are in the jar, right? So you have your kombucha and it contains the sugar, and then primarily the yeast will consume the sugar and make alcohols. Then the bacteria will consume the alcohols and make acids. So the yeast readily grow on the Petri plates, and so that's why we have been doing the spread plating and you have been counting, is because the yeast grow very easily and they're beautiful and easy to count. The bacteria are very, very finicky and hard to grow. So rather than grow the bacteria, we are instead measuring the amount of acid produced by the bacteria. So the primary acid, the bacteria produce maybe 3 or 4 different types of acid, but the primary acid produced is acetic acid, which is the acid in vinegar. Vinegar is acetic acid. This is why if you let your kombucha go for longer, it gets this kind of sharp, tangy vinegar smell because it's making a lot of acetic acid. Acetic acid is generally very antimicrobial. So the yeast and bacteria in your kombucha are acid resistant. So they can live in a high acid environment and it does not kill them. But most bacteria are not acid resistant and are killed by high amounts of acetic acid. So the way that the TTC test works is, let me actually, well, I'm going to switch to that page and then I'll come back to this. The way that the TTC test works is, let me get down there. Okay, so we are going to use E. coli, which is an intestinal bacterium, to measure the amount of acid produced. There's not normally a lot of acid in the intestine, so E. coli is very sensitive to acid. Acid will kill the E. coli. Okay, acid will kill the E. coli. Step 1 of things that you need to know. Step 2 of things that you need to know is the way that the TTC assay works is TTC is the reagent, and when the E. coli is normally living, it does electron transport and passes the electron to oxygen in aerobic respiration, making lots of ATP. When the TTC reagent is present, then the electrons will go to the TTC reagent, some of them, instead of going to oxygen, which will turn the TTC reagent red. All right, so the more red the sample tube, the better the E. coli are doing. So red is a measure of E. coli success. Since the E. coli are killed by the acid, the more red means less acid. Since the acid are made in your kombucha jar by the bacteria, acid is a measure of fermentation success. So if the tubes are really red or really red at all, then that means that the E. coli are surviving, which means there was not enough acid in your fermentation to kill them. So red equals living E. coli equals unsuccessful fermentation. Clear parentheses, absence of red, indicates no living E. coli, which equals lots of acid and tells us that your kombucha jar fermented successfully. Okay, so here are some pictures to hopefully help you understand this. And so this is what happens if you mix the E. coli with water and you don't use kombucha at all. The water will not kill the E. coli, so the E. coli will do respiration fine. Some of the electrons will go to the TTC, the tube will turn red. When you do, in this middle box, when you mix the pre-fermentation tea with the E. coli, the pre-fermentation tea has sugar. And so this will make the E. coli grow even better. Not only does it not kill them, it makes them grow better. And so your pre-fermentation will usually be a little darker than the red because the E. coli are growing really well. So they're giving even more electrons to the TTC reagent, making the reagent turn a darker red, and there's no acid because pre-fermentation. Okay. Hopefully, in your control jar, you have gotten a lot of acid production. So when you mix the control kombucha with the E. coli, it should kill all of the E. coli. The acetic acid produced by the bacteria should kill all of the E. coli. And so none of the E. coli should be able to pass electrons to the TTC. So the TTC reagent will stay clear instead of turning red. Sometimes these kombucha tubes will look like slightly yellow. That is the diluted tea color. So we don't care about yellow. We care about amount of red. Yellow is 0% red. Orange is some red. So we care about orange because it's secretly red. We don't care about any color that's not red. Okay, so hopefully this makes sense to you. The more acid produced, the less red, the more successful your fermentation was. Okay, back we go to week 8. Okay, so you're going to read all about this, and then you're going to take the Moodle quiz, which is going to ask you some questions about metabolism and like what happens in glycolysis, that kind of stuff. And some questions about the TTC assay. So even though we have just talked about the assay, definitely go back through the lab manual and read these sections because I don't want you to miss some detail on the quiz. Once you have finished the quiz, then you're going to work on the paper pre-lab. Okay, so let's go over to the Moodle and look at this week's paper pre-lab. It's like awesome, but a little tricky. So you're going to open it and it will look like this. And so the way that this works is every letter is the same answer. Okay, so in glycolysis, the 6-carbon glucose is broken down into 2 pyruvate molecules. Each pyruvate contains blank carbon atoms. So this is 3 carbons. Pyruvate has 3 carbons. And so everywhere that you see an A is going to be 3 after this, and so on. And so let's do the next sentence. This process breaks a bond, releasing electrons to the ready-to-accept electron carrier L, NAD+. So everywhere that you see L is NAD+, turning it into the with electron form B, NADH. So everywhere that you see a B will be NADH. Okay, so you do this whole page. Aerobic respiration is the first page and half of the back, or the second page. And then fermentation is the bottom of the second page. And so you'll just go through. Everywhere that there's a letter will be the same answer. So let's say that you secretly, initially, are like, is this, 6 or whatever, and then another place you come and you're like, oh, it doesn't make sense for A to be 6 there, then everywhere that there's a letter has to be the same answer. So you guys will fill this out. Hopefully this will coincide with you doing respiration and lecture and will be a good practice for you. So that's why we have this page. So that's what you're going to be doing for your paper pre-lab this week. When you get to lab, you're going to do the quiz like usual, and then you will set up the TTC assay. Okay, very, very important. The TTC assay, because it requires the metabolism of the E. coli, takes 45 minutes to sit there and do the thing in the little heat block, which is like a tiny incubator. And so, especially if you are in the morning lab, you have to hustle. Like, you need to know what you're doing and start immediately after the quiz, or you will not get finished before chapel. And so you need to have person A has to be really organized, but again, the rest of you, your lab will go more smoothly if the rest of you also can read and make sure you know what you're doing. And ideally, you will have communicated before lab about who's going to do which parts, so that as soon as you get there, you can just start. Because we are dealing with live E. coli in lab today, in lab this week, week 8. Everyone who is involved in the TTC assay will need to be wearing gloves, because we don't want you to get the E. coli on your hands. While you're wearing the gloves, absolutely do not touch your face. We also do not want you to get the E. coli on your face. So the big tip for this week is to get this going immediately. It takes 45 minutes. Once it is in the heat block and the 45 minutes has started, is a great time to do all the rest of the stuff that you need to do. So there are other things to do this week. These are all things that you guys have done before, so they should go really smoothly for you. The TTC assay is new, although you're excellent at pipetting now, so very good job. But the TTC assay is new, and it does take a really long time to incubate. So do be sure that you get it started straight away.